So thanks, Steve, for that introduction. And just to put you guys at ease, I will not do that, I promise you. All right, so just some disclosures. I feel obligated to always do this, just to let you know that I'm a scientific advisory board of DNA Nexus. I'm a consultant as part of the 1000 China Talent Program. Neither of these endeavors really are relevant directly, at least, to the research I'm going to present today. All right, so I'm going to talk to you about our work with respect to autism spectrum disorder. I'm going to start with kind of a perspective more broadly of trying to understand the genetic basis of neurodevelopmental delay, and then focus in on some of the more recent work that we've done with respect to autism. So I hope to convey to you, by the end of the day, some of our excitement with respect to the application of next-generation sequencing approaches, really to understanding what has been largely a black box, I would argue, for diseases such as autism, and really are beginning to open up quite wide right now with this application. Some of the work I'll present today will have really direct implications with respect to diagnosis, and hopefully I'll give you some, at least my vision in terms of how I think this will be important with respect to subtyping autism, and ultimately both behavioral and therapeutic interventions later on. All right, so first off, most of us, I think, have an understanding of what autism is. Most of the work I'm going to present today has been based on the old DSM-4 criteria, which is this triad of impairments involving social interaction deficiencies, speech delay, as well as repetitive behaviors. The CDC estimates a prevalence of about 1 every 68 births, at least in the US population. And it's important to realize, and I think most of us know this, that in fact the diagnostic criteria for definition of autism has changed over time. And so it's generally regarded as an autism or an umbrella disorder, and that umbrella has gotten bigger over the last, I would say the last 10 years. And so a lot of the individuals that wouldn't have been classified even 10, 15 years ago as autistic are now falling within spectrum. The other important feature is that there is significant comorbidity of autism, as most of you will know, with intellectual disability. I think the numbers are close to about 40 to 50% of the children with autism also have some form of reduced nonverbal IQ, epilepsy, as well as other neuropsychiatric conditions. So I'm going to be talking about the genetics of autism, not about other factors. And I think the data are pretty clear that there's a very strong genetic component for autism. These numbers have come down a little bit in recent years, but the estimate is currently a little bit excess of 50% in terms of the heritability of this. A lot of that data come from the study of identical twins, where if one twin has autism, there's a 70 to 95% chance that the other identical twin will have that diagnosis. And also from relative risk of really full siblings. So there it's 5 to 20%. So there's clearly, which is an order of magnitude above what the prevalence is in the population. The other feature of this disease, which I have some insight into, but not a definite answer, is something that we all recognize within the textbooks, is that there's a male bias toward this disease. So for the old DSM-4 definition, the estimated bias is 4 males for every female diagnosed with autism. And for Asperger's, which is now called high-functioning autism, that ratio jumps to 6 to 7 males for every female. This is not unique to autism. Many neuropsychiatric disorders have a male bias. Developmental delay, more broadly defined intellectual disability, those numbers are closer to 1.3 to 1.5 males for every female born. To summarize 10 years of genome-wide association studies that were done kind of from 2000 to 2010, basically relatively few recurrent common loci have been identified. So there was an idea early on, because the prevalence of this disease is so high, that maybe there would be a common variant that may in fact predispose to risk for this disease. There are a few that look potentially promising that may come up with much larger cohort sizes, but by and large, most of the early reports have not been replicated upon subsequent studies. We know from studies of Mendelian disease that there are clearly forms that are tracking within families, and there's about 8 to 10% of at least a genetic cause that can be ascribed to these kind of Mendelian searches for the disease. So that left people kind of circa 2005 starting to wonder, well, where is this genetic contribution? Where's this risk coming from? I'm not going to spend a lot of time about this, because maybe you've heard this from other folks around here, but my entree into this really came from the studies of copy number variation. And so I think one of the most important things that came in from really application of microarray technology around 2000 or so, was the identification from many labs that there was an excess of large copy number variants in children with developmental disabilities more broadly and autism spectrum disorder specifically. And so copy number variants, particularly ones that are large, that involve deletions or duplications of hundreds of kilobases, often megabases in size, were found to be initially tenfold higher, and later those numbers were readjusted to about a twofold excess of such large events in affecteds with autism compared to unaffected siblings. The interesting thing about this in our lab is that we got interested in this really from studying idiopathic cases of mental retardation. And we, as Steve mentioned, studied the genome, and we identified regions of the genome based on their organization that we predicted would be hotspots for large-scale copy number variation. So years before, I think, clinical microarrays became commonplace, we actually targeted those specific hotspots and looked at all the cases of idiopathic mental retardation cases that we get our hands on to see if we could discover new recurrent microdeletions or microduplications associated with developmental disability. So this turned out to be very profitable in terms of actually diagnosis. So back in 2006, this is one of the first recurrent microdeletions that we identified. We identified 4 patients that had, out of a group of 209 kids with unexplained developmental disability, we identified 4 that had recurrent microdeletions of a particular portion of chromosome 17q21. It's a region of about a half a megabase in size. There's about 6 genes in that region. And what we found in these families, when we identified this recurrent microdeletion, we never saw this deletion in controls. At the time we published the study, I think there were about 1,000 patients or adults or normal controls that we had looked at. And we found out of 4 out of 290 cases with developmental disability, we found this recurrent microdeletion. When we examined the inheritance of this, we determined that all of them were sporadic. So none of them were actually being transmitted from a parent. They were all de novo, occurring presumably in the germline of one of the parents. And when we lined up the patients and got photographs and clinical reports, it was clear there was very similar phenotypic features. So there was a facial dysmorphology characterized here by this bulbous nose, pronounced philtrum, this protruding tongue. These kids all had pretty severe to moderate impairment intellectually. And you can kind of see it from these pictures, but one of the interesting things from the clinicians, when they actually looked at these children, they realized that they actually had a very characteristic personality. They're very happy children in general, at least compared to their unaffected siblings. So this is now called the Coulon-de Vries syndrome. The gene was identified a few years later, but it was discovered first by this recurrent microdeletion. This is one that we discovered in 2007. So this is a recurrent microdeletion mediated by a hotspot at the end of chromosome 15. It's called a 15q24 deletion. Same kind of MO as the 17q21. Recurrent microdeletion, in this case, of 3 megabases, so 3 million base pairs that had about a dozen genes in it. All of these were sporadic or de novo, so that means they were not transmitted. And when we looked at the children, we determined that all of the kids, and I think this still holds today, there are about 25, 30 children that have been identified, at least in the literature, all of them have autism spectrum disorder. And they have very characteristic facial features. You can see here this frontal bossing of the forehead, this almond-shaped eyes, this elongated kind of head shape. What you can see from these pictures is that these kids are in the 25th centile of growth from pretty much their entire life. And so there's a very characteristic set of features due to this genomic imbalance of about 3 megabases of sequence that is sporadically deleted in one of the 2 germlines. Screening of over 25, now 30,000 controls has never found this event in the general population. The last example I show you is one that we published in 2008, and it's an intriguing one, and it actually helped educate us even further. So this was a recurrent microdeletion in the first couple cases that we saw. This is again on chromosome 15, mapping to chromosome 15q13. This is distal to the Prader-Willi-Angelman syndrome locus. This recurrent microdeletion, first couple cases were actually de novo. Then we got more families, and we determined that some cases it was inherited. So this, unlike these guys here, this could be about 50% of the time de novo, 50% of the time inherited. We initially identified these in children with intellectual disability and developmental delay. Later, a paper came out from Tony Monaco showing that this segregated with autism for 3 generations in a family, this particular deletion. Later on, papers in terms of schizophrenia reported this as one of the most significant copy number variants as a risk factor for schizophrenia in the general population. And later on, work we did with Heather Mefford showed that In fact, most individuals that had this, in fact, also had a form of epilepsy, so it was idiopathic generalized epilepsy. And when we screened a cohort of patients from Europe, we found that about 1% of idiopathic generalized epileptics carried this particular microdeletion compared to 0 out of like 3,000 controls from the same ethnic population. So this is definitely a high impact, high odds ratio risk factor with respect to disease, but it's in many cases not specific in terms of the outcome. So it predisposes individuals to a whole variety of phenotypes. These clinically, entire NIH institutes are built on different phenotypes here: epilepsy, autism, developmental disability. So this taught us that essentially this dosage imbalance, this recurrent microdeletion, could in fact manifest very differently. So work that we did over the last few years is we built a morbidity map of what we think are all the pathogenic CNVs by comparing very large numbers of cases versus controls. So scanning across the genome and finding excess deletions and duplications in children with developmental disability versus essentially adult controls. This is some of the data. We now have close to 30,000 cases, 20,000 controls, and the curve stays more or less the same. What I'm plotting on this curve is the minimum size of a CNV, either a deletion or duplication, as a function of a size. So basically, this is indicating here, if you just follow, let's say, pick 500 kilobases, what we're indicating here is that roughly about 25% of children with a diagnosis of developmental delay have a large CNV, 500 kilobases or greater, either deletion or duplication. And that compares with people in this room to about 10%. So 10% of you are carrying large CNVs, probably not directly relevant to any severe phenotype in you, but this differential we use to estimate or suggest that about 14% of the genetic cause, I put quotations around the cause, are the result of these very large CNVs in the human population. So if you think about that, right, developmental delay is estimated by some reports of close to 3 to 5%, so 1 out of every 20 births. This is a high impact in terms of actually contribution to disabilities, and this is why you guys run microarrays routinely for children that actually have developmental disabilities. You can break this out further by disease. So this was looking at all pediatric cases that have come in. You can see here is indicating craniofacial and cardiovascular, the best thought of as congenital heart defects. This is the same plot, just showing you the burden of CNVs. You can see it's much higher compared to the control line shown here in green. This is autism and epilepsy. So again, you can see this pattern of really elevated large CNVs in that population. And then these are pediatric cases with asthma, and there's in fact no difference with respect to the control line. So the important thing here is not every disease, at least pediatric disease, is related to large CNVs, but this is actually a great litmus test, right? This type of analysis for actually predicting which diseases do we expect to see de novo gene disruptive mutations in, sporadic or private, because that excess burden can't occur by chance. Last thing I'll mention, and then I'll shut up about CNVs. We've had the opportunity to then look at, this is data from about 2,500 families where a child was ascertained by developmental disability. I'm showing you the same plot that I showed you before, but what I'm partitioning out here are deletions versus duplications. And two other pieces of information: whether the CNV as a function of size was inherited from the father in blue, the mother in red, or was de novo. That means that it didn't occur in either of the parents' germline, and so it was sporadic. So a couple things you should get from this. There's a difference between deletions and duplications. A much larger proportion of the area here is de novo, and as CNVs get bigger in size, they're more likely to be de novo. Why? Because de novo CNVs, as they get bigger, involve more genes, are more likely to be either incompatible with life or incompatible with essentially production of additional offspring. But that difference is obviously dramatically different between duplications versus deletion. That goes back to old literature in genetics that says deleting a gene is usually more harmful than creating an extra copy of it. So we know that from Drosophila work. The other thing, which I think is the most interesting thing, if you have an incredibly good eye, you can see that the area underneath the curve for both the paternal and the maternal transmission is not equal. So it turns out, and this has now been reported now close to half a dozen times, if you look at the inherited events, about 58% of the large CNVs, defined here as greater than 400 kilobases in size, are being transmitted preferentially from the mother as opposed to the father. And for autism, that number rises close to 70%. So in diseases like autism and developmental delay, we're seeing mothers being preferential carriers of these large CNVs that are being transmitted as opposed to fathers. All right, so what did we learn from CNVs? A few things. We knew this actually from things like Down syndrome before, but we now have this actually over specific regions of the genome, that dosage imbalance is really important for contributing to disease, specifically diseases like developmental delay and autism spectrum disorder. And the other important thing was the importance of sporadic mutation. So for a geneticist thinking about a dominant mutation or a homozygous mutation that's being transmitted, these are mutations that are de novo in the human population, or they're de novo in the few generations back. Each of these accounts for a very small fraction of developmental delay or autism. So some of the examples I showed you are 0.1% of all cases of autism, or 0.2% of all cases of developmental disability. But collectively, they're quite common. So we estimate, as I mentioned, that 14–15% of intellectual disability can be accounted for these rare or private or de novo large CNVs, compared to 7–8% of autism spectrum disorder. Mothers are more likely to be carriers. That was an observation we initially made only with large CNVs, but I'll show you some data now based on more recent work. But CNVs have problems. And the problems, some of you may know, is that in many cases, the CNVs that at least have been shown to be pathogenic involve large regions of the genome, sometimes dozens of genes. And so you can't say specifically that one of those genes is responsible. In most cases, even the pathogenic CNVs, we don't know the gene that's responsible, which then limits diagnostics and therapeutics because you don't know the pathway that's being affected. In some cases, it may be multiple genes that have to be deleted to actually have an effect on a child. And still to this day, many CNVs are of unknown significance. We constantly get emails from people that say, I've never seen this CNV before, it's not in their literature, do you have it in your morbidity map? And the truth is that large CNVs generally have a much higher risk, like tenfold in terms of their increased risk in terms of developmental delay. But for specific CNVs, the actual significance for most is not yet determined. All right, so everybody knows about this. This is one of the two main revolutions, I think, in terms of human genetics, which is essentially next generation sequencing. So the idea that we could actually go back to all those patients and families where we hadn't identified a large CNV and look for essentially the same type of mutation, de novo gene disruptive mutations, became tractable when this technology came on board, and really the price of full genome sequencing dropped really by many orders of magnitude. And so the idea of actually looking at this and beginning to actually characterize these patients became tractable. So we were early adopters of exome sequencing, along with Jay Shinduri, and began to actually, instead of sequencing entire genomes, focus just on the protein coding portions, because those portions of the genome we can interpret, at least functionally, more readily than any other area of the genome. The genetic model was simple. We were going to take from the playbook of CNVs and say that what we're going to be looking for is now point mutations, single base pair substitutions that are de novo, that are killing genes, so loss of function mutations, nonsense, frame shift, and we're going to compare this in families where there's no family history of actually the disease. So work that we did over a period of about 3 years in our lab, collaborating with Debbie Nickerson, and in 2 other labs, that of Matt State and Mike Wigler, we sequenced about 10,000 exomes over a span of about 2 years, specifically from 2,500 families from the Simon Simplex collection of autism that had the following features. They had unaffected parents, mother and a father, one affected individual in that family, and at least in majority of the cases, one unaffected sibling. So the idea, we called these quads, was essentially to sequence mom, dad, affected child, and unaffected child, and look at the differences in terms of de novo mutations between this individual in the family versus the unaffected. So simplex families, no family history, not that that's a proof of no inheritance, but that enriches in potentially de novo mutations. And so the goal here was to use exomes to find specificity, right? To find the actual genes that are being broken de novo, because the idea would be see an excess of such mutations in children with autism compared to the unaffecteds. So I'm not going to go over those studies, and just to give you the summary, the take home from that. So what we and others determined is that when we looked at the rate of de novo mutations in protein coding sequences and classified them by type. So these are likely gene disruptive or loss of function mutations. These are synonymous changes, so they don't result in an amino acid replacement change. And this is the rate of missense mutations in affecteds versus unaffecteds. The good news, which has now been replicated multiple times, is we saw a twofold excess of gene disruptive mutations de novo, sporadic, in the affected individual compared to the unaffected sibling. If you compare synonymous changes, no difference in the mutation rate. And if you compare the missense, there's a slight but significant, borderline significant, but it is significant in terms of excess of missense mutations. These mutations are actually a focus of our lab right now because they might explain as much risk as these types of mutations with respect to autism. So we first saw this, we were quite excited. It was replicated pretty much by 2 or 3 other groups fairly quickly. So we were pumped. So the model from CNVs worked for exomes, and we now had specificity in terms of specific genes, which genes were actually being affected. There's always bad news. The bad news was, in fact, that when we compared our lists and our publications came out, we almost never saw the same gene hit twice. It was really sobering, I have to tell you. We did some back-of-the-envelope calculations, and the data were consistent that these were high-impact penetrant mutations, but just like there are multiple roads to Rome, there are multiple ways to get to an autistic state. We estimate now 400 to 800 different genes, when independently mutated in a family, will result in potential high-impact risk variant. So what does that mean? That means if you're a researcher like me, you're totally underpowered to actually discover genes. You just, even if you think you pat yourself on the back after 10 years of collecting 1,000 patients or families, that's nowhere close to where you need to be. We need orders of magnitude above that. If you're a clinician, what does that mean? It means if you specialize in terms of actually seeing cases with autism, and you've done it for 30 years, there's a good chance you haven't seen the same genetic cause of autism twice. So that should also be quite sobering, right, in terms of clinical. Some other things that a postdoc in my lab observed, and he actually saw this pretty early on, when he tried to determine where those de novo mutations that were coming from in these families, and this is a fairly small set of data, but it was highly significant, he found, based on looking at SNPs that were in close proximity to the de novo mutation, that 80% of the de novo mutations were coming from the paternal germline. So if mothers were more likely to be carriers of potentially pathogenic events, fathers are more responsible 80% of the time for these de novo mutations that occurred. And what Brian observed is when he actually looked at patients that actually had 3 or more de novo missense or de novo-like mutations in the protein coding, patients with 2, 1, or none, and plotted that against the paternal age at time of conception, there was a modest but significant correlation with paternal age. Work that came out about 6 months later, looking at full genomes, saw the same thing, obviously a much bigger landscape than exomes. And the estimate is now that slightly more than 50 new mutations exist in each child across the entire genome as a result of de novo mutations. But most of those are not mapping to protein coding areas of the genome. Interesting thing is, the data are all consistent, suggesting that 80% of these new mutations are coming from dads. And the older the father, the more de novo mutations you see in the child. And so I think the clock right now is set as about 2 new mutations for every year that a father ages. And that's thought to be the result of increased number of cell divisions that are going into production from mother spermatogonia to essentially sperm cells. So every 15 to 16 days, there's a cycle in males where there's a new mother spermatogonia that divides asymmetrically. One goes to produce sperm, the other goes to essentially produce another spermatogonia cell. And older men essentially will have offspring that have more de novo mutations. This is interesting with respect to autism. This is not restricted to autism. It's a property of all of us. So all of you are probably doing the mental math in terms of how old your dad was when you were conceived. But the bottom line is what's interesting with respect to autism, which is obviously an ascertained phenotype, beautiful epidemiological data from Sweden has shown that the risk of having a child with autism for fathers that are over 45 is about double. So 1 out of every 50 births compared to the general population, which is about 1 in 100, or a father at 25 years of age. All right, the second thing that we determined, this is work of an MSDP student in my lab, Nick Crum, was even though we had looked just at de novo mutations, now that we had exome sequenced everything, we could also look at inherited mutations. We could look at mutations that were potentially gene disruptive and bin them based on their allele frequency. So we could look at things at very low frequency, 0.2%, 0.1%, down to things that were private. So what does private mean? Private means we've never seen this mutation at this time in all the exomes that have been sequenced for controls and in no other family with autism. So in other words, this is a mutation that probably occurred in the grandparent or grandparental generation and is very specific probably to this family. So very, extremely rare, as rare as you're going to get. And he broke out the mutations with respect to whether they were loss of function or with missense. And then he just simply computed the odds ratio compared to the affected versus the unaffected in these families. And the real cool thing that Nick found is that if you looked at minor allele frequency of less than 1%, no difference. Odds ratios were pathetic. Minor allele frequency of 0.1%, that's pretty rare, no difference. When you looked at private, there was signal. There's a 1.15 odds ratio excess of these private loss of function gene-killing mutations that were being inherited to the proband compared to the unaffected sibling. And in fact, if you looked at the gene's tolerance to mutation, so we have measures to do that, these are genes that have almost no mutations in the general population compared to genes which mutate all the time, i.e. dead genes. What you saw is that this effect really spikes when you get to this kind of lower quartile or decile. So essentially, we have another hook, which is essentially these inherited but private loss of function mutations. And the really interesting thing, which I won't show you, if you ask the question from whom these came, which parent were these transmitted, almost all the effect came from mothers. So mothers, just like the CNVs, were transmitting preferentially to the affected sibling these loss of function private gene disruptive mutations, suggesting once again that mothers are preferential carriers of these loss of function mutations that are relevant to autism risk. The last point I'll make with respect to really the autism genes, even though we were totally bummed out that we never saw a lightning strike twice very often, there were a few genes, slightly more than a dozen, where we actually saw the same gene hit multiple times in autistic kids but never in unaffecteds. When we looked to see if they were related to one another in any way by gene expression or protein-protein interaction, what we found was that, in fact, the genes were highly non-random, at least in terms of their function. So whether it's whatever gene ontology database you look at, you'll get kind of similar results. Slightly more than a third of them map to essentially genes related to synapse function. So I should explain these diagrams. These are protein-protein interaction networks. So we're looking at, thinking at the concept at the level of the protein. So any line means that this protein interacts directly with this protein. If you see red, that means we found it as a de novo truncating mutation in autistic individuals. And de novo severe missense are shown here in blue. So synapse function makes sense. So these are genes typically as a group, glutamate receptors, genes important in terms of synapse plasticity, long-term potentiation. They're expressed highly postnatally, which I think is pretty interesting. And then they kind of subside later on in life. So this is stuff that's important in terms of the wiring of the brain in the first couple years of life. So we got a whole class of autistic kids, because the size of the circle represents the relative proportion of autism cases that we've seen with this. Then we have these other categories that I wish they weren't there, but they are the data. We had genes related to Wnt signaling, and some of you will know of Wnt signaling. Wnt signaling are particularly important in terms of cancer. They're genes important in terms of cell proliferation, and the signal is very strong coming from these. These are ones that we didn't anticipate, but these are genes associated with chromatin remodeling. So these are kind of, I call them master tinkerers, because they actually turn on early in development a whole slew of other genes, and they modify their expression levels, but they're not absolutely required for driving expression. They seem to like optimize essentially accessibility of chromatin. So a lot of chromatin remodeling genes in that set. Unlike these genes, which are postnatal, these genes generally have their highest levels of expression, at least in the developing brain, 10 to 12 weeks during postnatally, or I should say post-conception. So these are a much bigger target and more difficult target, we think, in terms of trying to think about potential therapies. If you plot all the loss of function mutations that we saw in those 2,500 families, 67% of the de novo LGD events actually project 1 to 2 steps away from this kind of core network of genes. All right, so this problem, it's a problem for you guys just as much as a problem for me. everything I showed you up to this point in terms of exome sequencing is just aggregate statistics. We see a twofold excess of loss of function mutations in unaffected versus affected, but I didn't tell you any specific genes. That's just an aggregate statistic of all the de novo loss of function mutations. And what you need and what I need is I need specific genes. I need to be able to say these are the genes that are mutated, actually result in an autism phenotype. And so which ones are they? Well, so you need two things to solve this problem. You need tens of thousands of patients. So not so easy, since the biggest collections were 1,000 or two back just a couple years ago. And the other thing that you need is you need technology that's actually dirt cheap to actually sequence these genes. So instead of sequencing all the genes, so just want to focus on a subset, maybe 50, maybe 100 genes. We want to test them in a large number of samples. So I had the good fortune of being a next door neighbor. Well, he's actually a floor below me or two, of Jay Shinduri, and he had developed this technology in part while he was at George Church's lab. If you haven't heard of it, you should read up on it because I think it's absolutely fantastic. But the idea is pretty straightforward. I won't go into great detail, but essentially once you have a set of genes that you think are potentially important and you want to test them in a large cohort, what you can do is develop these glorified oligonucleotides that have these complementary ends. They're called molecular inversion probes because they have two complementary ends. The distance between these is about 120 base pairs. You make thousands of them to correspond to the protein coding regions of, let's say, 40 or 50 genes. Put your patient DNA samples in essentially a microtiter dish, throw in DNA polymerase ligase, form a covalently closed circle, chew away with exonuclease the remaining single-stranded DNA, amplify with PCR, pull samples, and sequence. The power of this technology, when properly optimized, is you can actually sequence really thousands of patients for a reasonable price. So it costs us to resequence the protein coding regions of about 50 genes, about $12.50 per sample. So if exomes are poor man's sequence, this is poor man's sequencing to really go through a subset of candidate genes very quickly to find the ones that actually reach statistical significance. So this is work that's ongoing. We are targeting right now 205 candidate genes that have at least one likely gene disruptive event. And we have now submitted a paper under revision where we identified 90 high impact, what we now believe to be autism risk genes. So these are genes that are probably as close to necessary and sufficient for autism. About half of them are previously unknown and have no kind of association with autism spectrum disorder. As part of this work, I had to put together a collection of people that wanted to work together. It turns out in the autism field, this is not so easy. It turns out the researchers are kind of prickly, I think is the word I would use. And so I ended up flying to a lot of other countries to actually set up kind of deals and work with groups that actually had appropriate IRB consent to actually send us samples for actual sequencing. So we've managed now, the numbers are closer to about 17,000 patients that we put together. From now, it's more than 10, it's like close to 15 different centers around the world that are now being sequenced with these kind of targeted genes that we've identified from exome. The beauty of these is that a very large fraction, not all, but a very large fraction can be, once we've discovered something, we can actually go back to those patients and actually reassess the phenotype. And so this is work that we've done at the Autism Center here, both at Seattle Children's as well as the Autism Center at UW, is to bring patients back conditioned on a common genetic etiology. So we call this a genotype-first perspective because what we're doing is we're saying, okay, there's developmental disability out there and autism is part of it. What we're going to do is we're going to target some genes and we're going to target them deeply in a large set. We're going to prove statistical significance. And once we've identified 10 to 20 individuals with that mutation, we've never seen it in our controls, we bring those people back in, and then they spend 2 days here in our lab and other labs, and we have a kind of a jamboree focused just on studying this specific form of autism. So this includes the US, obviously, a lot of sites here, as well as more recently we've been able to expand in China. Getting the diagnosis of autism running in China has not been trivial, but we actually have a few labs that are doing this and doing this quite well. So we just had our first paper with the Chinese actually published yesterday. All right, so what's the benefit? So as I mentioned, once you get to double digits or even close to a double-digit number in terms of cases, and you have done a large number of controls, you bring those patients back in and you re-phenotype them. So this is just some of the statistics. This is an early paper that I published with Rafe Bernier, 13 loss of function mutations based on screening of 8,000 cases. 0 out of 8,792 controls. So these are loss of function gene disruptive mutations in this chromatin helicase DNA binding protein 8. When we go back to the families, 11 out of 12 of them are de novo. So just like our CNVs, we're getting high impact, probably necessary and sufficient, or as close to it, genes as you can imagine. And then when we assess the phenotype, and remember we screen broadly developmental disability children, 87% have a diagnosis of autism, 50% have intellectual disability, and interestingly, you can see it from these pictures, 80% have macrocephaly, defined here as more than 2 standard deviations with respect to head circumference. Thing that we totally didn't anticipate, but when we actually brought these kids back in, 73% of them had severe gastrointestinal dysfunction, defined here as actually weeks of constipation followed by weeks of diarrhea that would last usually for the first 10 years of their life and for some reason abate around puberty. The other thing which has not reached significance yet, but I believe will, is sleep dysfunction. Close to 70% of these children have very severe and almost idiosyncratic sleep dysfunction. The first 2 parents that came in describing these children on the top said that, here in Seattle, said that their kids, and they didn't know each other, and these are obviously de novo mutations, so they're not related, that their children would go for 3 days in a row without an iota of sleep. And one woman said to the clinicians is that she would lock the doors at night because she was falling asleep and she was afraid her child would wander away. And so she actually locked them in their room because she couldn't stay awake with them. So they would do this for periods of time and then crash and then reboot, so to speak. So we had the opportunity to make a mouse, not a mouse, but a zebrafish model. We were able to knock down the gene using morpholinos as well as CRISPR-Cas. This is just some data that was in the Cell paper where we actually did measurements of the optic testa, the distance between them in the zebrafish. And we saw what appeared something that was reminiscent of macrocephaly, kind of unusual for a knockdown in zebrafish to see this kind of overgrowth of the head. When we stained with markers that would stain neurons versus other types, this was all due to an expansion of neuronal anterior as well as midbrain neurons. So an excess of neurons seems to be relevant to this disease, at least for these kids. The other thing that we did, we actually had the ability to look at once we made the knock out, or knock down, I should say, zebrafish, we could stuff them with their mouths with fluorescent beads and measure the movement of those beads along the GI tract. And we can show that, in fact, there's a delay in actual passage of those, significant delay. So they have a gastrointestinal dysfunction. If you stain for neurons, which is what you're seeing here with the controls versus the CHD8 knockdowns, there's actually a reduction in the number of neurons in the zebrafish when we knock down. So in the brain, excess of neurons, in this case cortical neurons, and in the gut, a deficiency of enteric neurons. This has just been replicated in the mouse in a paper that came out about 2 months ago. This is DIRC1A. I don't know if anybody's ever heard of this. I didn't until we started seeing lots of kids with mutations in it. It turns out this is a very interesting gene. It's a dual tyrosine phosphorylation regulated kinase that is thought to be one of the 2 genes, the other gene is DSCAM, on chromosome 21, that is responsible for the dysfunction associated with Down syndrome. So people have been studying this gene for a long time, but they haven't actually found patients that actually had loss of function. So when we started doing exome sequencing, we started to see de novo loss of function of this gene. We did the same thing. In this case, we screened, when we published this, 7,000 cases versus 8,000 controls. Eventually, all of them were de novo except one. In this case, the kids had 100% ID, 100% microcephaly, 90% had late onset epilepsy, 83% had autism, a particular severe form, which is impaired expressive language. And the comment that came from the clinicians when they brought the photographs together was that there was a characteristic facial dysmorphology. So in addition to microcephaly, they have a very characteristic pointed chin, which you can kind of see in these children. This one individual doesn't. She's actually had reconstructive surgery. This is so severe, this kind of high nasal bridge, this pointed chin, that these children, when their adult teeth erupt, they actually have to have them removed because there's not enough space in the developing jaw for those to essentially come in. This is a very specific feature associated with a very specific mutation in a gene. Would never have been seen, I don't think, unless you had actually pulled out those patients and separated them from idiopathic autism. We don't have to make a mouse model because a mouse model was made because of the implications with Down syndrome. And in the mouse model, the mouse actually have reduced brain volumes, and the hets actually have problems with anxiety as well as pre-weaning disturbances with respect to their behavior. This is the last example I'll show you. This is a gene that we know almost nothing about. It's an activity-dependent neuroprotective peptide. It was actually studied years ago because of its potential role in therapy for Alzheimer's disease. So it produces a very short peptide that is thought to be, could be protective for Alzheimer's. So these are the statistics. This is clearly a high impact, we believe, variant. 9 out of 9 that we've tested within families, these numbers have gone up, are essentially de novo. ID and ASD, 90% of loss of expressive language. These children have neither micro or macrocephaly, but if you look at their heads, you see something. You see this kind of high anterior hairline and this almost frontal bossing of the forehead. It's an overgrowth specifically of the cortical region of the developing brain, not a global increase. It's fairly reproducible, and it's been seen in pretty much the majority of the patients that have come in that have these types of mutations. We don't have a mouse model here, but people have made a mouse model, and when they knock it out completely, you get a failure for the neural tube to actually close and premature death. HETs have neuronal glial pathology and reduced cognitive function. So we think this is, once again, a high impact example of a gene. So coming to kind of a close, what do we know about the genetics of autism, kind of from microarrays all the way to exome sequencing? Well, we know some pretty interesting things, and this is all data based on those 2,500 families. We know there's a significant excess of large CNVs in autism probands. A lot of these are de novo, but when they are transmitted, mothers are more likely to be carriers. Okay, so these are typically event sizes greater than 100 kilobases. There's an excess of de novo gene disruptive mutations, point mutations, so single base pair substitutions that are actually resulting in nonsense frame shift. These occur 80% of the time in the paternal germline. And then finally, we have an excess of gene disruptive point mutations in conserved genes when transmitted. Those are going preferentially from mothers to their affected sons. We see almost nothing in terms of respect to females, although our numbers for a number of females with autism are still relatively few. The last point I'll make is we think, based on some back of the envelope calculations, that we now can explain about 30% of the genetic risk, or at least 30% of the risk, or the risk for 30% of the patients that in fact have a simplex autism, so no family history, primarily by these large CNVs and these gene disruptive events. So where's the rest? Well, this is more of a pie in the sky perspective, but we believe the rest will become, at least some of it will become apparent with full genome sequencing. And this is some preliminary data from a postdoc in my lab, Tishel Turner, who has actually done full genome sequencing. So now we're moving away from exomes and now doing full genomes. I know a lot of clinicians don't like the idea of moving from exomes to genomes because we're just moving into a lot of uninterpretable landscape of the genome. But we've begun to do this. We specifically focused on families and individuals that were negative by exome for at least a likely gene disruptive event, and they were negative with respect to at least the CNV analyses that we had at the time. And what we've seen, and this is not to be considered proof yet, we've seen an enrichment of de novo and private smaller CNVs over regulatory elements, so enhancers and promoters. Specifically, we see this over genes which already have a prior with respect to autism. So in our 53 families, we found 16 families that had high impact candidate events, including 9 in autism risk genes. And we suggest a possible diagnostic yield for autism on the order of about 15% on top of exomes, on top of CNVs. And just to show you some examples and to show you how complicated this may be, here's an example of a family where we have an affected male. He has 2 very large deletions which should have been detected, you would think, by microarray, but they were not. And they occur in genes that are already risk factors for essentially autism. These CNVs are essentially not seen in the general population. Inherited one from the mother, one from the father. This individual has a small deletion of 35 kilobases within a known developmental delay gene called MBD5. It's a methyl DNA binding protein. It also has, I think, the second reported mutation in DISC1. This is a gene that's actually been implicated very strongly in terms of schizophrenia. I think 3 or 4 generations in a Scottish pedigree. And then this is an example of one where we have 3. I'm just showing 2 of them, a deletion in a gene that's well known for developmental delay, ARID1B, but again, doesn't affect the exons. And a deletion just immediately downstream of probably the second most important gene with respect to de novo mutations, SCN2A. It's a sodium channel gene, which we think takes out regulatory elements. At least the data would suggest from ENCODE that takes out regulatory elements in those. This is a tough nut to crack, and I think probably will be the more common form as opposed to these monogenic high impact, where you have multiple events coming, sometimes de novo, sometimes inherited from different parents, and they're converging on children, almost like a burden of these types of disruptive events. So in summary, I've given you kind of a view of the large copy number variance, the fact that we now think that 7 to 8% of cases with autism can be accounted for at least part of the risk by these large events. And I define large here by typically hundreds of kilobases in size. For pediatric cases with development delay, that doubles, so it's about 15%. So the sequencing of protein coding regions has been fantastic for giving us candidates. It also has been sobering in terms of the realization of how many different ways there are to get genetic mutations associated with autism. If we estimate 500 genes, we now think we've got a handle on 90 genes. Most of this has happened over the last year or two, largely because of the advent of next generation sequencing technologies. And so obviously these genes, once proven, I think statistically, become candidates for diagnostic, right, for screening patients more routinely. We consistently see this maternal carrier bias for large CNVs, smaller CNVs, and now point mutations that break genes. And that just seems to be a general property of essentially mothers of children with autism. There's many ways to think about that. Two ways that we think about it is that it may be that a mother or a female requires a greater burden to actually manifest autism, so they can actually more likely be carriers. Or alternatively, they have a phenotype that we don't clinically recognize as autism, but it's something else. So it's obviously very interesting and important, I think, to go back to these risk factors that are being transmitted from mothers and to assess the phenotypes of those individuals. So combined, these gene-killing mutations, whether they're CNVs or point mutations, we think are 27 to 30% of patients. And I've shared with you this idea of a genotype-first approach. This is not really that new. It's the same thing that works for cancer, is the idea that simply having a diagnosis, whether it's autism, autism spectrum disorder, or if it's developmental delay, is totally and woefully insufficient. What you need to know is you need to know the root genetic cause of it. And so for those families that you identify that have a common cause, our data suggests that there's much more similarities in the phenotypes of those individuals. And I would argue that if there are therapies, whether they're behavioral or pharmaceutical, you wouldn't want to treat children all the same. I think the idea that you would treat a child that actually has autism because it has an underproliferation of neurons, i.e. DYRK1A, the same way you would treat a child that has an overproliferation, is just ridiculous. And I have a feeling that some of the therapies that have already been developed probably worked in the first trials because they had maybe less heterogeneity in those initial samples, but when they applied it to a bigger cohort where they had in fact many more causes, the actual trials failed. So this is obviously the theory behind all the precision medicine discussions that are going on. This is our patting ourselves on the back slide. This is where we were in 2007. This is where we were in where we think we are in 2016 in terms of explaining the genetic risk of autism, with most of the emphasis being on private and de novo gene disruptive mutations. But I don't want to give you the impression that means that we've identified all the genes here. These are aggregate kind of calculations in terms of risk for the different types of variants. So we still have a big challenge, which is to find the individual genes, regulatory elements, and proteins that are being affected. This part has become totally apparent to me. We need, as a community, and this is the clinical, researcher, and families, become much more integrated. The idea of having fiefdoms of 100 families or 200 families that you spent 6 years collecting is totally irrelevant. We should be actually combining all these resources, doing things systematically, and having the ability to go back to those patients once we identify the mutations. So this is definitely a research and development aspect. And some of my clinical colleagues say, what is the point of telling a family that their child actually has a CHD8 loss of function mutation when there's absolutely nothing you can do for them at this point? And I would tell the clinicians in the room that think this way that they're thinking too narrowly. Because what I've seen from the CNVs all the way down to the case of now having individual genes is that once you identify 10 or 20 families with a specific genetic lesion that you can tell them with pretty good confidence, in fact, is relevant to the disease phenotype, those families interconnect. And once those families interconnect, they share a lot of information, much more than a researcher or a clinician will ever have, and in fact come up with some very interesting and sometimes practical solutions to management of the disease as opposed to trying to treat it. So I think there's great benefit to defining genetic subtypes for the families so that they can understand not only the cause, but they can connect with others, which will have much more similarity in terms of genetic etiology. I can't tell you how many times mothers have said to me, she goes, my Johnny was never like all those other kids that I went to in the support group, but now that I've met 4 more families that have this type of mutation, they are just like the same playbook. I think this is really important, and I think it's actually underestimated that how powerful those will be going forward. And the last thing I'll mention, and this is the promise, right? So we have all these genetic subtypes, and it might seem bewildering, and we still have most of the genes yet to discover. But once we identify the individual subtypes, obviously that's going to be valuable for diagnostics, because once we have like CHD8 or DYRK1A, we can now say something to families where we find those loss of functions de novo. But I actually think that the solution down over here in terms of therapeutics is actually understanding how those proteins work and how they particularly work with each other. So while there may be hundreds of genetic subtypes, I think in the end this is going to boil down to dozens of protein and gene expression networks. And I think the treatments, whether they're pharmaceutical or whether they're behavioral, are going to be done down over here. So as geneticists, we live up here. But what we need to do is we need to actually move down here much more, because there's so many of these genes which are just a name right now, like ADNP or even CHD8, that we really need to understand the biology, just like people have done 25, 30 years ago with FMR1 and Fragile X syndrome. We need to understand the biology before we actually come up with therapies that make sense going forward. So I'll close there. These are all the folks that did all the work in my lab. The highlighted ones particularly did a tremendous amount. Highlighted work of Brad Coe, Nick Crum, Holly Stessman, who's just recently accepted a faculty position, and Brian O'Rourke, who's actually at Oregon as an independent investigator. Great colleagues, Jason Dury, Debbie Nickerson, and Rafe Bernier in psychiatry, who actually is on the front lines seeing the patients. And then this is just a partial list of what's close to probably 2 or 3 slides of all the clinicians that I've worked with on this kind of massive endeavor to kind of coordinate and sequence and target sequence and recontact families. So thank you for your attention. I'm happy to take questions in the last 5 minutes.